Good evening,
Good
ma'am.
evening, sir. Good evening.
Yes, ma'am. Good evening. Shall I start, ma'am?
Yes, sir, you can start.
Yes, ma'am. Thank you so much. Hi, and welcome to the wonderful session of the biological classes, my dear students. All of you welcome once again the biology NEET classes. Now we are going to discuss about the application of biotechnology part 2. Okay. Here, first we are going to discuss about nematode resistance in plants. You know about not only the plant involved in the biotechnology process. Some of the animals like nematode, it has a specific gene structure. The gene structure is also, it has the ability to resistance in plants. That means animal cell resistant to in plants. Okay, that is very, very important, my dear students. It is also another part. See there, one by one. Several nematodes parasite. Parasite, what is the meaning of parasite? Already you know about. Host is also alive, isn't it? Parasite, saprophyte, symbiosis. These are the three types of, what is that? Hetero nature, isn't it? Parasite, nothing but the host is also living in nature, and also it's live along with the host. Such type of organisms, such type of plants are called as parasites. The example they have given as a fungi. Okay. And also saprophyte. Saprophyte is nothing but the dead organic matter. Take an example, say any one of the plant or animals after dead. Okay. Such conditions, the some of the microorganisms, they will grow upon it. Such type is called a saprophyte. And the last one is called a symbiosis. Symbiosis means both. Okay. Symbiosis means give and take policy. Take an examples of lichen. Likewise, some of the nematode is also resistant in plant. See here, several nematodes parasite a wide variety of plants and animals, including human beings. They have given an example. See that a nematode Melodigyne incognitia. Melodigyne incognitia infect the roots of tobacco plant and causes a great reduction in yield. That is very, very important. Which plant it will affect, you know? So one of the nematode, it will affect the tobacco plant, tobacco. Okay, in our board. Here there are 60 alkaloid present in even in a single piece of tobacco plant. Already studied in tobacco, where drugs. Okay, that category. Already studied in the plant. Plant causes a great reduction in yield. Okay, so some of the nematode, particularly, they ask the question, in this particular nematode, what will happen, it affect the entire yield. That is very, very important. Okay, sometimes they may ask the question, tobacco plant, okay, the production will be completely reduction by the nematode. Name is called as. Likewise, they may ask the questions. Okay. And the next one is a post-resistance plant. What is my post-resistance plant? And RNA interference. Post-resistance. Post-resistance after. Okay. So after the production of any one of the plant product, it will also fight against many diseases. Such category is called as post-resistance plants. Okay. See that the art of gene slicing is a part. Art of gene slicing. What is the meaning of slicing? Slicing is nothing but a small fragment. We may made a cut. Okay, it is a very good art. Just like what is that? So many art is there. So, see here. The RNA interference, the simply called as RNAi. Okay, RNA interference is the term given to the phenomenon inside the living cell by which short RNA molecules control the gene expression. That is very, very important. What is the main rule of that? That RNA interference gene, it will control the gene expression. It will suppress the gene expression. It will not allow the gene to, what is that? Express itself. Okay. Such type of gene is called as, what is that? RNAi. So otherwise called as RNA interference. Interference means what is prevention. Okay, that is very, very important. So don't forget, my dear students. Here, once again, I repeat for you. Nematode is also has the properties fight against many resistant in plants. Example, what is that? Melodiu gynae, incognitia. Infect the roots of the tobacco plant and cause a great reduction in yield. So these are the animals affect the harvesting of the plant. Okay. So next one is a post-resistant plant and RNA interference. RNA interference is nothing but one of the genes present inside the living organisms. What they will do, it will control the gene expression of others. That is very, very important. See the next one. The RNA interference, also known as the gene slicing, is a cell regulatory mechanism to control the amount of protein being produced or the type of protein being produced. It acts at the level of RNA. See, this is the main important. As a gene slicing, what is the meaning of gene slicing? RNA, it controls the activity of other genes, whether they are RNA production or protein production, all those things. This concept, this reactions, genes, expressions, RNA interference genes, it was first studied by the Andrew Fire and Greg Mello. So these are the two scientists that identified these genes. Which one, So the gene is called as, what is that? RNAs, slicing genes. So RNAs, okay, RNAs, ribose nucleic acid interference, simply called as interference. That's why, okay, this was first studied by the name of the scientist, Andrew Fire and Greg Mello is the name of the scientist. Okay. See, the RNAs takes place in all eukaryotic organisms as a method of cellular defense. What are the main activities? The cellular defense, it acts upon the cellular level. That is the important point. This method involving cell slicing of the specific mRNA due to complementary of double-stranded RNA molecule that bind to and prevent translation of mRNA slicing. Here, what happened, the RNAs act upon the specific enzyme, site of mRNA, messenger producing RNA. Why it is act on the mRNA, you know? The mRNA essential for the production of translation process or protein biosynthesis. Without mRNA, there is no any protein biosynthesis takes place. At the same time, what happened, you know? The complete translation process completely stopped. So the RNAi, okay, involves slicing of specific mRNA due to the complementary double stranded RNA molecule. Then only it bind each other. Okay, complementary means, you know, it will be exact join with the nitrogen base of the double stranded RNA as well as the mRNA. Okay, this can be, what is that? Prevents by the RNAs. And another one is using agrobacterium vectors. Agrobacterium, already you know about this. This is very, very important. Agrobacterium is one of the tumefacians, tumor-inducing protein molecule secreted by the one of the vector is called as agrobacterium. Is mostly in the plant. Okay, ground call effect. All those things already we studied. Using agrobacterium vectors, nematode specific genes were introduced into the host plant. So this is also another one. And next one is, see that these two RNAs being complementary to each other formed a double stranded RNA. Which one? mRNA as well as RNA. Both are compound contact close together as a result of the formation of double stranded RNA molecule that insert the RNA and the slicing the specific mRNA of the nematode. Okay, nematode. When it joined these two things, which one you know? The two RNAs being complementary to each other formed a double stranded RNAs that initiated by the RNA interference and the slicing of the specific mRNA of the nematode. The consequences was that the parasite could not survive in the transgenic host, expressing specific interfering RNA. The transgenic plant therefore got itself protect from the parasite. So that is the main aim of the RNAi. Okay, so once again repeat for you. So what are the two RNA molecules united together? What are they? The mRNA as well as double stranded RNA. How it will the double stranded RNA molecule formation takes place? You know, the mRNA bind with the RNA interference. So as a result, you will get double stranded RNA. That is given here. See, double stranded RNA. That is the initiation process of the, which one you know, slicing of specific mRNA of the nematode. Okay. As a result, what happened? No, the consequence was that the parasite could not survive in a transgenic host expressing specific interfering RNA molecules. The transgenic plant therefore got itself protect from the parasite. Okay, so go to the next one. The molecule contributing to RNA interference are already we discussed microRNA, otherwise called as mRNA is already we discussed. Messenger RNA we thought, isn't it? But it is not a messenger RNA. It's nothing but the microRNA. Micro means it's a small fragment like structures. It is a very small small RNA molecules. Okay. Another one is siRNA, small interfering RNA. These are the two RNA molecules involved in this process. Okay. So what are they? MicroRNA, simply called as a mRNA, a small RNA molecule. Another one is called as a siRNA, small interfering RNA. These are the two molecules essential. See that what they will do? miRNA are small RNA molecules, about 22 nucleotides. So how many nucleotides? Sometimes they may ask the question in the NEET point of view. How many nucleotides present in the mRNA? Okay, how many nucleotides present in the mRNA? Likewise, they ask the question. So clearly you should note it down. How many? 22, 22. Along that are encoded by the genome. mRNA consists of small molecules, but it contains 22 nucleotides is there. Long that are encoded by the genome. It acts as a post-transcriptional regulatory by binding of complementary sequence on target mRNAs and thereby usually resulting in the blocking of translation and gene slicing. When it act upon it, what happens? Automatically the translation process completely stop. That is very, very important. Okay. So this is very, very important matter. Post-transcriptional regulator. It is also called as post-transcriptional regulator. By binding a complementary sequence, complementary. So then only what happened, the process, the translation process will occur. Okay. Suppose it's bind with the microRNA, what happened, the process completely blocking. See that the blocking. So that is very, very important. Blocking of translation and gene slicing process. So what happened? The protein, it will not reduce. That is very, very important. So the mRNA, the RIRNAs. See the next one, the siRNA, small RNA, small interfering RNA also has the same properties as a miRNA. The only difference being the origins. siRNAs are formed from the degradation of double-stranded RNA carried out by the enzyme dicer in the cytoplasm. This is also another important point. Okay, which one? The siRNA. Okay, how it will be act? Okay, on the double-stranded RNA with the help of the one of the enzyme. That enzyme name is called as dicer. Dicer means two. Note it down, my dear student. This is very, very important. Without dicer, the siRNA, it will not function. Okay, see that. The RNA interferes induced in mammalian cell by introducing antisense RNA to generate double-stranded RNA structure which are cleaved, to generate the small fragment RNA. See that here, the RNAs and siRNAs. The siRNA, what happened, it will not function without the dicer. That enzyme is called as dicer. The enzyme present in where, in the cytoplasm of the cell. Cytoplasm. Okay. After that, what happened? The RNA interface. Okay. Introducing the antisense RNA to generate double-stranded RNA molecule structure which are cleaved to generate the small fragment of RNA. These two are. Okay. The dicer, it will split into a simple molecule, double-stranded RNA molecule split into a fragments. The double-stranded RNA are identified and cleaved by the dicer enzyme to generate a small interfering RNAs or microRNAs, or mRNA is a single-stranded RNA molecule that inhibit the translation of mRNA, whereas the siRNA or double-stranded RNA molecule that cleave RNA. Here you must note it down. Two things is very, very important. Which one, siRNA, miRNA. siRNA act upon only with the presence of the dicer. Okay. When those two are activated upon the RNA molecules, it inhibits the translation process. Okay. The mRNA, siRNA are double-stranded RNA molecules that are cleave RNA. So these are important things. This is the concept you cannot able to understand properly. Okay. You go through it, you will get the ideas. Okay. First of all, you should note it down. siRNA is there. Okay. The siRNA without a dicer, it will not act upon because it needs some enzyme. That the enzyme is called as dicer present in the cytoplasm. Okay, first point. Second one is double-stranded RNA are identified and cleaved by the dicer, already we discussed. As a result, what happens is generate small interfering RNA or microRNA. Okay, the microRNA in single-stranded RNA molecule that inhibit the translation of mRNA. There's a single-stranded RNA, siRNA, inhibit the translation of mRNA. That is called as, what is that? MicroRNA. Whereas siRNA are double-stranded RNA molecule that cleave the RNAs. Okay. See that. A riboprotein complex is generated by the small RNA fragment called RISC. So this is another one. This is the important point, my dears. RISC. What is my RISC? RISC. RNA-induced slicing complex. RNA-induced slicing complex. Sometimes they may ask the question in the form of expansion. Okay, which bind to the homologous mRNA molecule that initiate and cleave the slicing the gene expression. Okay, it will completely what happened now? It will not express. Suppose when it full molecules, full polynucleotides, then only it will be expressed. Suppose if it is cleaved or sliced by means what happened? No, it will not express itself. That is very, very important, my dear. This was done by the one of the riboprotein complex is called as RISC. That RISC is otherwise called as RNA inducing slicing complex. The RNA induced slicing complex. In mammalian cells, RNAi effect can be introduced by generating small interfering RNA, that is siRNA or small hairpin RNA, shRNA. See how many types of RNA is there here? See, siRNA, mRNA, okay, and also double stranded RNA, and also a small hairpin RNA. See the next slide, you can easily identify. Okay, see that double stranded RNA molecule. Okay, this double stranded RNA molecules, they act upon the dicer. What happens? It gives a SIR. After that siRNA formation, what happened? So undergoes a RISC protein inducing dicers. It bind with this one, so it act upon it. As a result, it will be splicing. Okay, it is splicing like this. After that, what happened? No, the RISC also comes out. Finally, you will get this type of what is that? mRNA molecules. See that one? So some examples of GM crabs, genetically modified crabs. See that first one is examples called as plant, maize, tobacco, maize, and also brassica and nabas. Maize, molecular product right or introduced. What are the molecular products right introduced? Maize, the herbicide resistant or high lysine. Lysine already in our body is one of the amino acid, basic amino acid. Tobacco is a plant, the herbicide resistant. Again, maize, so the malaria, influenza, vaccine, and cancer. Molecule produced, triads introduced. Brassica nebus, oil-free form, glucosinolate, uric acid. So these are the importance of what is that? GM crabs. What the next one is, my dear students? Applications in medicines. That is very, very important field in nowadays. It's a very important and most vital part of the biotechnology or genetic engineering. Okay. The applications of in medicines. The recombinant DNA technology has enabled mass production of the therapeutic drugs. In our report, nowadays we are facing so many problems. Okay, in 1984 onwards, AIDS. Before that, what is that? Cancer. Nowadays, in our report, dengue. Okay, and also another one, hepatitis B virus. Now, COVID-19. So we need some a revolution or new therapeutic drugs. Okay, see, so in this part, the genetic engineering is play a vital role. See that. Recombinant proteins, some of the recombinant protein have been, what is that? Tabulated here. See here, medically useful recombinant product. Some of the product are recombinantly made. So it is used for the medicinal aspect in many application format. They have listed here in the form of what is that? So tabulation format. First one is blood clotting factor. Just earlier about that, but blood clotting factors. How many factors? 13 factor is there here. The blood clotting factor 2 has given here. Okay, 8th factor and 9th factor. So what is the application of this one? The replacement of the clotting factor missing in the patients with the hemophilia A and B. Already you know a little about. So one more is that is called as 11th factor is also missing here. But except 11th factor, there are 2 factors also involved in the hemophilia A, hemophilia B. The hemophilia A is responsible for the clotting factor 8th. And B is responsible for 9th one. It is the one of the therapeutic medicinal usage. Next one is a tissue plasmogen activator, simply called as TPA. Tissue plasmogen activator. Dissolving blood clot after heart attack and strokes. Sometimes what happen, some of the person suffering from heart attack. Okay, inside the bloodstream, there is a clot is there. That clot is completely removed. So dissolving of blood clots. How will you, with the help of the one of the recombinant product is called as TPA, is called as tissue plasmogen activator. Don't forget, my dear. Okay, and then next one is erythropoietin. Just in the heading is the answer. Is that heading is also has answer. Erythropoietin, erythropoiesis, simply called as RBC. See that application. Stimulation of the formation of the erythrocytes, RBC, red blood corpuscles, for patients suffering from anemia during kidney dialysis. Okay, a side effect of the AIDS patient treated by the drugs gene cloned in the tobacco. 3 diseases have been reported here for the application process of erythropoietin. So one of the recombinant protein molecules isolated in the laboratory by genetic engineering and biotechnology aspect. What are the diseases? First one is anemic. When during kidney dialysis, because kidney is not properly functioning, that time what happened already you know about in zoology, isn't it? And also it is a part of side effect of AIDS patient treated by drugs. And one more is the gene cloned in the tobacco. These are the 3 points, is very, very important, my dear. And the 4th point is what is a platelet derived growth factor, simply called as PDGF. What is that? Platelets derived growth factor. So you know about it. It is very, very important. Okay, stimulation of wound healing process. Normally, the wound healing process also done by the one of the factor. Platelet derived growth factor, platelets. It is also essential for the blood clotting mechanisms. And the next one is very, very, very important one. It's nothing but the interferon. Okay, the interferon mostly medically very, very important one, because it is treatment of the pathogenic viral infection and the cancer. You know, well, but recent year also they were used for COVID-19. Okay, BF17. So interferon is an antiviral protein molecules. It's fight against many pathogens of the virus. That's why they were produced in the laboratory and also used as the application part. And then human insulin. Already in our human insulin, humulin already studied. It's also isolated from the recombinant technology of pig and human. Okay, the pig insulin component of the polypeptide is similar to that of the human being after some physiological and chemical changes which occur in the nucleotide sequences. After the modification, the pig insulin molecules act as a human insulin. See that one? The treatment of insulin dependent diabetes. That is very, very important. And the next one is human growth hormone. Human growth hormone is very, very essential for their growth. Replacement of the hormone in short stature people. Some of the persons, you know about it. Due to growth hormone deficiency, the persons are any other people. So some of the people you can see it's very short in nature. Okay, dwarf due to the genetic disorders, due to the lack of the growth hormone. Suppose that persons we can introduce the growth hormone, the person will be what is a replacement of hormone in short stature people. So these are the important things. Next one is, my dears, calcitonin. It is also very, don't forget, my dear student, this tabulation, don't forget it because the question should be here. Calcitonin, treatments of rickets. What is by rickets? Rickets. The bone, what is that? Softness. Calcitonin is also very important. And chorionic gonadotropins. This is also very important. Chorionic gonadotropin. Gonads secrete the one of the hormone is called as chorionic gonadotropin. It is very essential for the fertility. Okay. Due to the lack of fertility, what happened? You know what happened? It will not fuse with the egg. It will not get the child. So such cases, they were used chorionic gonadotropin hormones. It is also one of the proteins. Treatment of infertility. Okay, this is also another one. Simply, vaccines. Vaccines means there are so many vaccines have been identified or invented. Prevention of infection disease such as hepatitis B. It is similar to that of the cancer. It is similar to that of the cancer. Herpes, influenza, okay, pertussis, meningitis. What is the meaning of meningitis? Is nothing but the in brain. What happened? You know, meningitis, syphilis, meningitis. The water molecules stored in the brain, etc. These are all controlled by the vaccine preparation by biotechnology. And interleukin, it is also very important. The interleukin is also very important, just like a interferon. Interleukin, interferon. Don't forget, my dear. Modulation of action of immune system. Both are interrelated. Already told interleukin and interferon. Interferon, antiviral protein molecule secreted by the laboratory. Likewise, interleukin is also one of the important protein. It is also used for the modulation action of the immune system. Next one is alpha lactoalbumin. It is also very important. Okay. Nutritive protein in milk of Rosic cow. Some of the cow, the protein molecules, the protein content will be very less. Such category, we can enrich the alpha lactoalbumin in their milk due to the technology. Already you know about that. You have to isolate the specific gene from the Rosic cow and identify, and another one, you know about the foreign gene, otherwise called as allele gene, is also responsible for alpha lactoalbumin production, very high amount. Both are incorporated, and finally what happened? You will get the high nutritive protein in milk of Rosic cows, particularly alpha lactalbumin. Next one is alpha AT, alpha antitrypsin. Alpha antitrypsin, simply called as alpha AT. Gene introduced in cows, sheep, treating emphysema, a natural antibiotics. It is also one of the antibiotics. Emphysema, it is nothing but the swelling. Okay. Some of the cow or buffalo or sheep, what happened, you know, due to some diseases, what happened? Emphysema, it is nothing but the readiness. Okay, redness of the body part anywhere. So it can be prevented by the introduction of alpha AT, otherwise called as alpha antitrypsin. Next one is lysostaffin. The lysostaffin gene introduced in cow, antibacterial activity. Because the cow or buffalo or whatever in veterinary science related, you know, all the animals, they are affected by so many bacteria and virus diseases. Okay, mouth, food, and so on. So many skin, rashes. Okay. Likewise, that conditions we have to introduce the new protein molecules. That protein molecule is called as what is that? Lysostapin. Lactoferritin. Okay. Gene introduced in cow prevent mastitis. Mastitis already is no very about. Once a particular person suffered by a cancer, okay, any part of the body, okay, it is transferred from the one place to another place by blood. Okay, it can be easily transferred. Okay, when it is transferred throughout the body, the condition is called as mastitis. So the cancer spread throughout the body by blood as a liquid connective tissues. Okay, it can be prevented in in cow. Which animal? Cow. Lactoferritin. Okay. Next, the spider silk protein molecule. The spider silk protein. Gene introduced in goat produce a ultra strong medical industrial microstructures, tennis racket stirring. Okay, it's one of the diseases. Okay, caused by some of the bacteria. Due to this one, we have to use a spider silk protein. Again, lactoferritin, antimicrobial used to treat GI tract infection. GI means gastrointestinal tract infection, gene cloned in the tobacco and potato. Okay, tobacco and potato, both are plant. Both are considered getting known as Solanaceae family. Potato, Solanum tuberosum. Okay, so that is very, very important. So the both are same family, but the gene is cloned. It is fight against many diseases. The gene is called as, then gene produce the protein molecule is called as lactoferritin. The lactoferritin is act as antimicrobial activity. That is very, very important, my dear students. These are important points. Don't forget, my dear students. So in this part, the question is there. Okay. So go to the next one is, say that genetically engineered microbes. So not only the protein molecules produced by the genetic engineers, and also some of the microorganisms also produced by the genetic engineers. What are they? What are the main functions of this one? See, the E. coli, Escherichia coli, simply called as E. coli, Rhizobium meliloti bacterium. And next one is Pseudomonas fluorescens bacterium, Pseudomonas putita bacterium, Trichoderma fungus. So these are all very, very important genetically engineered microbes. So here you must note it down. Three things is very, very important. First one is E. coli, Rhizobium, Pseudomonas putita. That is very, very important. The last one is Trichoderma. Trichoderma is the one of the fungus. It induces the skin diseases. Suppose when you are wearing a wet cloth, inner wear, moisture cloth, as a result, what happened? It will induce the small patch like red patch like a structure in your skin, and some irritation is also there. Reddiness, irritation. Due to that, which one Trichoderma. It's a fungus attack. Okay, see that one. So genetically engineered microbes. See here, E. coli. Already everybody is the most important vector, and also microbes used for the biotechnology as well as genetic engineering. Okay, mostly. See here, what are the main functions or application of this E. coli bacteria? The production of the human insulin and human growth factor, interferons and interleukin. Already I told you, the interferon, interleukin, and also human growth factor. And the last one is called as insulin. These are the four important components also genetically produced in lab with the help of the vector is called as Escherichia coli, otherwise called as E. coli. Okay. And the next one is, what is that? Rhizobium meliloti. Rhizobium already mostly found in the leguminous plant. What is the main functions of the rhizobium in plant roots? Nitrogen fixation from the atmosphere to soil. Okay, see here. Nitrogen fixation by incorporating NIF gene. What is the meaning of NIF gene? NIF gene is nothing but nitrogen fixating gene. That's all. Okay. In cereals crops, cereals, you take an example of all the cereals. Okay. But the rhizobium meliloti, the one of the bacterium. Okay. What is the main role of the bacterium in plant? Maximum the nitrogen fixation, particularly it incorporate with the NIF gene, nitrogen fixating gene, and also it enhances soil fertility. Okay. And the next one is Pseudomonas fluorescens. Take an example, fluorescens. It's also glittering bacterium. When you are examined under the microscope, you can see it. Pseudomonas fluorescens. It is also present in the soil. We can isolate by the process called a serial dilution process. Prevalence of frost damage in the plant. Example, strawberries on which it grows. Prevention of frost damage to the plants. Okay, in strawberry, already it's one of the fruits. Mostly it's a high nutritive substance. Vitamin C is mostly found in it. Okay, and then fourth one is very, very important, my student. Without this one, there is no any NEET question. Pseudomonas putita, already we discussed in the intro part. Scavenging of oil spills by digestion hydrocarbon of crude oils. Okay, so normally about that. When the ship is moving from one country to another countries, in between there is some natural disasters happen. Immediately what they will do, they will pour the crude oil into the sea. As a result, what happened? The tide's speed will be reduced. So the ship is escaped from the large tides. Okay. So after that, what happened? The crude oil present as it is in the waterbed. As a result, so many microorganisms, millions of aquatic animals, they will not get proper light as well as the oxygen molecules. They will lead to die due to the crude oil deposition. So that case is what they will do, the Pseudomonas putita, it spray in the form of powder form. Spray it on the surface. As a result, what happened? The Pseudomonas putita, the complex molecules of the crude oil, it will be split into simple molecules, carbon, hydrogen, oxygen, nitrogen, like that. It can be exhaust from the atmosphere. As a result, the ozone layer, ozone layer is clear. The aquatic, it will be escape from the oil pollution. Next one is trichoderma fungus. Already we discussed it. Trichoderma fungus. Production of enzyme chitinase for biocontrol of fungal disease in plant. That is very, very important. Because why? Production of enzyme chitinase, because the fungus cell wall is made up of chitin. Already discussed it. It acts upon only the chitinase enzyme. Only the chitinase enzyme, they can split the molecules of fungus cell wall made up of chitin. So the chitinase is enzyme. Chitin is a substrate. The chitinase is the enzyme. So it acts upon substrate enzyme complex. Okay. For biocontrol for fungal disease in plant. When you are using this one, what happens? Some of the fungus, it will not affect the plant. It controls the fungal activities. Okay. These are the very, very important, my dear students. Don't forget Okay, so what are the things is the blood clotting factor, 8th and 9th, tissue plasmogen activator, TPA, erythropoietin, platelet derivative growth factor, and interferons, human insulin, human growth hormones, and calcitonin, chorionic gonadotrophin, vaccines, interleukins, alpha lactoalbumin, alpha antitrypsin, lactostapin, lactoferritin, spider silk protein, lactoferritin again it is used for the GI and tract infection. Okay, and then genetically engineered microbes, you know about the 5 have been given: J. coli, Rhizobium meliloti, and Pseudomonas fluorescens, Pseudomonas putida, and also Trichoderma. Okay, so go to the next one, my dear student. This is very, very important. How the genetically insulin molecules are produced. It has a specific technique. Then only the commercial available insulin molecules we can produce. How they will produce, we will see one by one. Insulin used for diabetes, already in our body, isn't it, my dear? Yeah. The insulin used for diabetes was earlier from pancreas of slaughter cattle and pig and caused some patient to develop allergy. What is the meaning? Slaughterhouse, you know about slaughterhouse, you know, what is the meaning? You know, cow or buffalo, where there is a place to cut the cattle or pig or sheep. That place is called a slaughterhouse. During that process, what happened? No? Some of the pancreas secrete the insulin hormone. That pancreas is also isolated from that pancreas. They isolate the insulin hormone. That insulin, it is not match all the human beings. Some of the person, they will get allergy. So such cases, what they will do, will not do any precautions. So the insulin consists of 2 short polypeptide chain, the chains A and B chain. What are the 2 chain is there? No, there are 2 chain is there. The 2 chain, what is that? Chain A and chain B, that are linked together by disulfide bridge. That is very, very important, my dear students. So many times they were asked the question in this part. Okay, already we discussed these things also. The insulin, it contains 2, what is that? A and B chain. The 2 B chain and A chain is also connected with the help of the bridge-like structure, disulfide bridge. See that one? I will show you how it will be now. See that one? Take an examples of insulin molecules. Okay, so this is considered here chain A, isn't it? Think about that one. Another one is considered as a B. See that one? So this is this one is called as A. This one is called as chain B. Okay. In between, see that this is the another part, isn't it? This is the another part of the another part. Okay. Already you know about one side. This is the protein molecule. Both are protein molecule. What is that? Insulin. What is that? Molecules. Insulin. INSULIN. Insulin, isn't it? Insulin molecule. Where the secretion is there, already in our board. Liver. Okay. The pancreas is there. Pancreas. Okay. Pancreas. Okay. The pancreas, it has a 2 cell is there. What are the two cells? One is called as alpha and beta and three cell is there, and also gamma cell is also alpha and beta and gamma cell also. So alpha cell secrete, what is that, glucagon. Glucagon. Okay, glucagon. So what about the beta cell? It secrete insulin. Okay, insulin. Okay. Insulin. Okay. So the glucagon, what is the main functions of glucagon? You know about glycogen. What is that? Converted into glucose. Isn't it? Glucose. But what about insulin? The reversible reaction. Glucose converted into glycogen. Okay, this process. Why I am telling these are all comes at the beginning, But here the alpha chain as well as the beta chain secreted by the beta cell, Langerhans pancreas beta cell insulin. In between the disulfide. Okay, disulfide means, see here, so there is a S. Okay, double bond, again there is a S. Yes, sulfhydryl bond, SH bond. Okay, sulfhydryl, sorry. See this bond, I will mention different color, then it will be better for your understanding, isn't it? See here, SH bond is that, sulfhydryl. Sulfhydryl bond is there. Okay, SH. It is a bridge between these two, just like a nitrogen basis. Okay, SH. Okay, so this is the bond. Okay, this bond is called as which bond? Sulfhydryl bond. Here, why I am telling the chain A and B are connected by the sulfhydryl bonds. So disulfide. Okay, that is very sulfide bond united the two chains. Okay, that is very important. Okay. See then, the chain A and B that are linked together by disulfide bridge. In mammalian, including humans, insulin is synthesized as a pro-hormone. Primitive stages, they will not produce the mature hormone. They produce as a pro-hormone. That contain an extra stretch is called a C-peptide. Don't forget, my dear students. Sometimes the insulin also, it has a extra stretches is also there. That stretch is called as, what is that? A C-peptide. The C-peptide is not present in the mature insulin and is removed during maturation into insulin. It is not present in the mature insulin. Don't forget, my dear student. So already we discussed there is a disulfide bridge is there. The disulfide bridge, what happened, when the disulfide bridge is present in the insulin molecule, that insulin molecule is called as proinsulin. Okay, not only that, and also it has some extra stretches also present. The extension is also there. Suppose if it is a mature insulin molecules, that in insulin molecule, they do not have the extra stretches. Okay, so that is called as a mature insulin. So proinsulin converted to mature insulin by the removal of the C-peptide. Okay, that is very important, my dear. Don't forget, this is also a important point. See that concept booster, you should note it down here. What are the amino acids there? This is also very, very important. Don't forget, my dear student. This is very, very, very important. Insulin pod. There are two types of chain is there. Chain A has 21 amino acids. Chain B has 30 amino acids. Chain C has 30 amino acids. So total, how much is that? 81. So 81 amino acids there. The insulin gene has a 153 nucleotide and it is present in the chromosome 11. So don't forget, my dear student. Okay, don't forget, my dear student. Insulin gene has a 153 nucleotide. Okay, 81 amino acids and chromosome number 11. Insulin is a dimer of A chain and B chain linked by the disulfide bond and is composed of 51 amino acids. Just you join these two. So these two, 30 plus 21, how much is there? 51 amino acid with a molecular weight 5,808. Okay. So this is very, very important, my dear students. So up to this level, okay. So we will finish it up to this level today, this class. Remaining things we will see the next classes. Okay, my dear. See, what are the things? I will summarize all those things. So today, what we discuss, nematode resistant in plants, several nematode, particularly the Melidio gyne incognitia, infect the root of the tobacco plant and cause the great reduction in yield. This is the first point. Not only that, and also some of the post-resistant plant and RNA interference is also one of the genes. Okay, it controls the gene expression. How it will be controlled the gene expression means the RNA is bind with the mRNA. So the mRNA as well as the RNA is also complement to the double-stranded RNA. It splices into single fragment. As a result, what happened? The gene expression was controlled. Okay, that things already we discussed. Okay, this was first introduced by the name of the scientist called as Andrew Fire and also Greg Mello. Okay, so this is very, very important. Now, RNA takes a cellular defense, which vector they were used for the identification of this one. Acrobacterium vectors, nematode specific genes were introduced into the coast plant Acrobacterium using Acrobacterium vector nematode. Vectors used for the nematode specific gene were introduced. Some of the hybridization, what we did, E. coli used as a vector, isn't it? Here we used which one? Acrobacterium vectors used for the nematode specific gene isolation and also proliferation. Okay. So what is the main uses of this one? First of all, you should note it. There are two types of interference sources. One is called as microRNA, otherwise called as mRNA, or small mRNA molecule is there. The siRNA, small interferon RNA molecule is also there. It has a nucleotide mRNA, otherwise called as miRNA. It has a nucleotide 22 nucleotide is there. Use the post-transcriptional regulator and blocking the translation of gene slicing. Already we discussed that part. Isn't it? So, and also the siRNA is also active with the help of the enzyme is called as dicer. The dicer is present in the cytoplasm of the cell. Then only the translation process occurs. Okay, so that is also already discussed. And one more is the one of the riboprotein molecules complex is generated by the small RNA fragment is called as RISC, simply called as RNA inducing slicing complex. Okay, what they will do, the RISC which bind to the homologous mRNA molecule and initiate the cleavage, slicing the gene expression. Okay, that things also already we discussed. Next, okay, what is that? So some examples of GM crabs. GM crabs, genetically modified crabs. So many crabs is there. Here they have chosen 4 only: maize, tobacco, and also brassico nebus. And then what is the main applications in medicine? It's a very, very important, my dear students. Don't forget, my dear. So application in medicine is also very, very important things. Okay. See that what are the things already we discussed? The blood clotting factor, 8th and 9th, tissue plasmagen activator, erythropoietin, platelet derivatives, and interferon, human insulin, human growth hormone. Okay. and also the calcitonin, chronic gonadotropin, vaccine, interleukin, alpha-lactalbumin, alpha-AT, lysostapin, lactoferritin, spider silk protein, and lactoferritin. These are all very, very important things. Genetically engineered insulin, already you know about the insulin molecule consists of two chains. One chain is called as A and another chain is called as B. These both are united with the help of the bridge-like structure with the disulfide bridge. Okay, the pro-insulin contain what is it? You know, there is a and B chain peptide, not only that, and also the C peptide along with the insulin molecule. Suppose C peptide present in the insulin molecule, it is considered as a pro-insulin. Okay. In mature insulin, the C peptide is not present. It is absent in the insulin molecule, mature insulin. And insulin molecule contain what are the components? It is very, very important, my dear students. Don't forget this one. Chain A has 21 amino acids. Chain B has 30 amino acids. Total A and B containing 51 amino acids. Already we discussed here. C chain containing 30 amino acids. Insulin gene has 153 nucleotide and it is present in the chromosome 11. Don't forget, my dear students, how many chromosomes we are having? 46 chromosome is there. The 11th chromosome is insulin chromosomes. The insulin is a dimer of an A chain and B chain linked by the disulfide bond and it is composed of the 51 amino acids with the molecular weight 5808, my dear students. That's all about, my dear students. Today class is over. We'll see the remaining things on the next classes. Thank you so much for the participation. Thank you. Bye. Take care. Good night. Thank you, ma'am.
Thank you, sir.
Thank you.